Welcome to the Georgia Initiative for Climate and Society, Getting to Know Ourselves, to Know Us seminar series. And we have this every single month on the first Wednesday of the month. And we are pleased today to have Dr. Nibbelink talk about modeling vertebrate species responses to climate change and sea level rise. I forgot to introduce myself. I am Susan Warnell. I'm on the executive committee of the initiative. Our director, Dr. Yeager, is taking a leadership class today. Dr. Nathan P. Niblink's research focuses on modeling mechanisms that influence the spatial patterns of animal distributions on the landscape and how these models can be integrated into GIS-based tools to help manage our ecosystems. Specific to the Center for Climate and Society, Dr. Nibbelink utilizes spatial analyses tools to evaluate effects of sea level rise and climate change on species habitat. A variety of species distribution modeling algorithms are employed to evaluate range shifts in species in response to projected climate change. The Nibbelink Laboratory also focuses on creating mapping tools to facilitate decision making in the face of climate and other landscape changes. Excuse me. Please help me welcome Dr. Nibbelink.
Thanks for coming. I will try to get done in time. This is a new amalgamation of projects for me, so I'm not quite sure how long it's going to take, but I'll promise to try to be respectful of your time. Today I'm going to focus largely on the aspects of work that I've been involved with historically and am again moving into now that deal with vertebrate species response to climate change and some of the secondary effects that vertebrates then have on the ecosystem. So I'll move right into this in the spirit of the Getting to Know Us series. I just slapped up the majority, I think almost everyone in my current lab here, with the sort of quote-unquote climate sea level rise team on the top. And we have a whole variety of projects going on looking at animal movement patterns in the landscape, particularly at metrics of fragmentation, animal movements, and habitat structure, and how those changes are influenced by climate and land use. So in the spirit of getting to know you, shouldn't have changed it quite yet. I'm going to cover several projects I've been involved with and not go into too much detail in every one of them, but try to give you a flavor of the kinds of models that we use and that are being employed to look at vertebrate species response to climate change and some of the effects of climate change. So I'm obligated to throw out the Mauna Loa graph, as I'm sure several people have. And I wasn't quite sure what I was going to say about it because you guys all know this already. So I decided, I had an inspiration from a seminar I saw last week to go and Google, in Google Scholar, climate change and species distribution. And then I rescaled the CO2 change, which isn't really fair, with number of articles, and recognized that we must be able to get a handle on species response to climate change because in 2002 we exceeded the CO2 with the number of articles coming out on. I know this is totally meaningless, but it's kind of a jab at people doing these sort of Google things. I'm not sure exactly what that means, but I did limit it to actual articles as opposed to citations. So anyway, at least I made that correction. So I'm going to start with some historical work I was involved with in the early 90s after I graduated undergraduate. I became a research scientist, an assistant research scientist. I was a technician at the University of Wisconsin, which eventually led to my master's program. And I was hired to work on this project, and I basically became the modeler as the graduate student, David Hill, left his PhD to pursue other things. And so I spent most of my time on this project actually running the simulation models and generating the new distributions, which I'll show you in a minute. So this project was potential effects of global climate change on small North temperate lakes, physics, fish, and plankton, and I'm just listing collaborators and funders there. So I just want to talk briefly about fish, and this will also apply to amphibians, which will be coming up later. They're largely ectothermic, and therefore climate is expected to have a big influence on their metabolism, their breeding, their foraging, and specifically to lakes, because fish have thermal tolerances and thermal preferences. The habitat volume in that lake, when the waters warm up, might have some influence on fish use of that habitat. We're lucky in that these lakes are well studied, and we know a lot about the physiology of these organisms, and so we have a lot of mechanistic information to go on. What I'm showing here is just simply a depth profile of the abundance of particular species of fish. I think in this case it's a cool water fish gill, and then I show temperature and chlorophyll profiles on that plot. So we know what the fish are doing. Likewise, we know what the plankton are doing. My collaborator, Bart Pastasio, did a lot of work in Rhode Island and in the North Temperate Lakes, looking at zooplankton and their diurnal vertical migration. And so we had a lot of measurements in these same set of lakes as to what zooplankton were doing at noon and at midnight in response to their environmental stressors, which included metabolic response to the ambient temperature at different depths, predation pressure, etc. So we decided to employ a modeling approach that started with a selection of general circulation models. I'll move on and I'll just list them there. I won't go into a lot of detail, but I'll just provide sort of a reference. And of course these models produce air temperature, wind speed, precip, solar radiation, and humidity, which feed into this dynamic reservoir simulation model. Also required for that model is an ice breakup model, so we need ice breakup date and the light extinction coefficients for the lake, and these did remain constant in the runs that we did, even though these things might feed back. We did not model those defects. So quickly, here's just a picture from the DiResin website, which still exists. I'm not sure how frequently it is still used, although I found several recent articles that have employed it. I'm sure it's been improved upon over the years. So out of DiResin, we get water temperatures, mixing depths, ice-free seasons, and then we can take fish thermal niche information, feed that into the zooplankton diurnal vertical migration model, and look at the changes in fish and zooplankton distributions after calibrating the model to current conditions. This is the only equation I'm going to put up in the entire seminar, and I'm not going to go through it in detail. But we used a dynamic modeling approach to modeling the zooplankton diurnal vertical migration and their selection of the optimal habitat in which to reside. Basically, the dynamic modeling equation is backfit to current fitness based on reproduction, increment to current condition or size, and the probability of survival, each of these across each time step. And the idea with dynamic modeling is that you solve it through backward iteration in order to yield the optimal trajectory and then ultimately the optimal thermal habitat or depth to select in the lake based on some of those ambient pressures, which are all modeled in functions under each of these different model terms. So what do we find? Moving right on. With physics, we found with lake stratification, these plots show depth and day number and temperature profiles. And so basically, with the baseline scenario on the top and several different GCMs represented in scenarios below, we see longer, in general, across all of our simulations. I'm only showing you a sample. We saw longer stratified seasons, stronger thermoclines being established, and a deeper epilimnion. So the warmer temperatures are going deeper, but that metalimnion habitat is becoming constrained, and it's generally getting deeper, warmer, deeper, depending on the strength of the stratification, and that certainly varied among lakes. So what are the fish doing in response to this? Well, we saw in general, again, although it varied among simulations, in general, we're increasing habitat for warm water fish species, decreasing habitat for both cold and cool water fish species because of the deepening of the warm waters and because of the sort of constraint of the cool thermal gill. So what did the plankton do? Again, these are these depth profiles showing chlorophyll and temperature for Sparkling Lake, one of the North Temperate long-term ecological research lakes. And right over here we have the optimal depth prediction from the baseline scenario, and then a doubling of CO2 scenario over there on the right. I put in this reference line just so you can kind of see what's happening. We get about a 2-meter shift deeper in the mode of the fish thermal preference habitat, and we get about a 2-meter drop in where the large zooplankton are hanging out, and particularly Daphnia pulex, which are dominant in the lakes that we did this with. And then we also see about an 8-meter increase in depth of where the little guys are at night. So, summarize this work briefly. We saw increased surface water temperatures, deepening of the thermocline, a shift in that fish thermal habitat, a shift in the diel migration behavior of zooplankton, and so the conclusion is that climate change might have important implications for the interactions of predator-prey and their food resources, which is not necessarily a surprise, but in 1993 was one of the first articles to address this in lakes that I was aware of at the time. I didn't go back and do another thorough lit review of it. But we thought it was pretty cool at the moment. And I did a brief search for current articles. I was happy to see that a colleague of ours at Wisconsin, Steve Brandt, has done this recently for the southern basin of Lake Michigan and found a very similar result where you get a tightening up of that thermocline, a reduction in particularly the cool water fish gill habitat. So those results are persisting in some systems. Okay, so we learned a lot from this exercise. Can we apply these approaches to other species? The problem, of course, with a lot of other species is that we lack a lot of the nice detailed mechanistic information that we had available as kind of a luxury for this project. So increasingly, management plans are mandating strategies for adaptation and mitigation to projected changes due to climate, sea level rise, etc. So what tools do we have at hand to deal with these kinds of problems? If we're lucky, we have some literature on preferences. A lot of species have been studied at least a little bit, although the recent project on some herps, boy, there are some where you can find almost nothing. And then if we're luckier, maybe, we also have some observations of sites that are occupied by certain species. So what do we do with that? We are generally doing species distribution models. And I'm going to go into a little bit of the different types of species distribution models that are being used and provide a couple of examples that we've used in the lab. What is species distribution modeling? It's been referred to in a lot of names. Element distribution modeling, which comes from the heritage programs because they refer to the locations of species not as individual observations but as element occurrences. Niche modeling, modeling habitat potential, species habitat modeling, and there are probably many more. Sometimes used interchangeably, and sometimes authors are fairly specific about what they mean when using those terms. The important take-home message from this is that the use of the term is widespread, but Pearson published in 2007 saying that it's somewhat misleading since what we're actually talking about most of the time in respect to the types of models I'm going to show you is the distribution of suitable environments is what's being modeled, not the species distribution per se. That's important to keep in mind. So there are two general approaches that have been used, both deductive and inductive approaches. Deductive approaches, of course, are where conclusions are developed from a combination of premises. This is based on a priori ecological knowledge, and then we, from the literature, or from personal observation, and then we do a spatial expression of that information using the layers that we have available from either habitat for the species or, in the case of climate change, the different climate variables that we have available from GIS data sets. And then that's what we call a typical suitability model, where we'd stack up those layers and be able to produce a projected distribution based on the kinds of changes that we would see. An inductive approach, of course, where conclusions are developed as an extrapolation from available data, from the bottom up, so to speak. It's sample-based, quantitative, often statistical, and it's what most of us are call modeling, of course. So, what exactly are we modeling? I find this useful just to keep in mind, even though it's not a, I don't see this as a hard thing, but it's a useful framework. Morrison and Hall talk about the species range as the limits within which a species can be found, and that considers only broad geographic space. Species distribution, they refer to as the fundamental niche. So it's a spread or scatter of species within that range, and it considers geographic space and those environmental components like elevation, rainfall, etc. And then the habitat level, they refer to as the realized niche, which of course is that combination of resources that promote occupancy, survival, all the life history traits. And then, of course, at the finest level, we have our species observations, we hope. Generally, SDMs are operating at this intermediate level, the level of the distribution in response to environmental characteristics. To varying degrees, I think they go different directions depending on the type of algorithm you're using and the amount of mechanistic information you have available. So, as an example of a deductive modeling approach, I'm going to jump from the past to one of the newest projects we have going on in the lab, and that's assessing vulnerabilities and adaptive capacity in coastal communities. This is a project with whom several people in the room are collaborators. Bob Cooper, Clint Moore is here, Kyle Barrett got a job at Clemson, go Kyle. And then we have two new PhD students on this project, Elizabeth Hunter and Rachel Guy, and they've had a lot of influence on early aspects of this work. And the example in this show is something Elizabeth put together last spring and this fall. So, in modeling vulnerability, there are generally three components that are being considered. Sensitivity of the habitat or of the species to the types of changes, the exposure or the risk presented by the types of changes that are going to occur, and then, of course, their adaptive capacity or the ability, perhaps, for us to manage for the kinds of changes. And so that's just sort of providing a basic framework for how we're looking at vulnerability. We're particularly interested in identifying habitat features that correlate with occupancy and abundance of the species, and ultimately the individual PhD projects will get involved in some of the higher level mechanisms of animal movement and their ability to move between patches and experience nest survival in the case of birds, etc. Particularly also we're interested in landscape factors, connectivity, patch size, and edge density, the kinds of things that particularly in a coastal marsh environment we're going to see changing with sea level rise. So, moving on to assessing exposure, of course we're interested in the temperatures, the climate variables, although with the range of what we're looking at, we don't see necessarily including those per se in some of the modeling exercises. But particularly also we're interested in sea level rise, and I'll go a little bit more into what we're doing there right now and what direction we're going with that. So, we had to select a suite of species to use for this project. We're focusing on species of conservation concern. We're prioritizing species listed by more than one southeast coastal state. And for birds, we're also considering the partners in flight listing. Also, ease of monitoring. Obviously, there's a logistical constraint because we're planning on implementing a monitoring program that we hope will persist long term. And we also want to represent diverse natural histories and diverse sensitivities to the types of changes that we're going to see. And that's specifically the example that I'm going to show you gets into that. So, We are using SLAM 5, so those of you who are familiar with SLAM 5 or SLAM 6 that is in progress, beware that we're not relying too much at this point on absolute predictions and some of the problems that we see with SLAM 5. What we're really after are what kinds of changes are happening in terms of patchiness and what kinds of responses will we see with the vertebrate species that we're considering. And we, as of this morning, probably have just begun a collaboration with Clark Alexander, so we'll be working with him on the SLAM 6 results as they come available. Okay, so I'm going to show you as an example of the deductive modeling approach, this assessment that Elizabeth put together again last spring and this fall, where we looked at some of the SLAM habitat categories and did a thorough literature review, and we also have to thank our undergraduate helper, Laura Menjak, who did a lot of the lit searches on this. And we identified for 28 species, 29, the types of habitat that were suitable for nesting and foraging across all of these species. And here's two examples, the clapper rail and the Wilson's plover, where we identify the habitat. Those are the SLAM categories for that particular habitat type. And we're calling those species fundamental niches. In addition, we're looking at change scores with the progression of sea level rise to a 1 meter level according to the SLAM 5 predictions. So we're looking at the change in total area, which is the total number of habitat cells, the patch size, the mean number of cells or habitat patches that form adjacent cells, habitat quality, which is the ratio of high and low quality habitat, and then habitat permanence, which is a fraction of cells that were also considered habitat in the prior time step. So what did we see? This is for clapper rail. We're looking at the left at total nesting area in this case, and on the right, total nesting area quality, stratified in quantiles from high, medium, and low quality, largely for clapper rail based on elevation because they nest at higher elevation, or higher or more intermediate? Sort of intermediate because they want to stay away from avian predators but be high enough not to have their nest inundated. So if we go through about 4 time steps here, we can see, as expected, and as well known from the SLAM 5 results, SLAM 6 has a little more marsh accretion, so it won't show quite the dramatic amount of change, but I still think from the preliminary results Clark showed us when he gave the Warnell seminar a few weeks back, that some of the patterns of change are going to remain the same. And this is, I think, the last one right here. You can also see some of the new habitat forming inland, but in general, the overall quality is defined by this exercise is declining. This is just a blow up of the savanna area, because most people in Georgia are familiar with what Tybee and Wausau and Skiddaways in there look like, and so I thought it would hit home a little more to see some of these again, even though we don't expect the magnitude to be quite so great. Okay, so just plotting briefly across years, the index, a rescaled index of habitat quality, patch size, total area, and permanence. We can just generally see for a clapper rail that each of these is declining over that time period as expected. But of course, across all 28 species, we get a whole variety of different kinds of responses depending on their sensitivity. So, salt marsh habitats experience the most change across the 4 metrics. Species that use salt marsh extensively, like rails, sparrows, diamondback terrapins, were ranked the highest for exposure. Future studies should use SLAM 6. I inserted this one. Elizabeth prepared a couple slides for me, and I took a great look at these. And a greater understanding of the species habitat relationships at a finer resolution, which again is what the students will be working on. And this will allow us then to stratify by sensitivity and select candidate species to monitor, because we're not going to monitor all 28 or 29 species that were in our original selection. That, in combination with a power analysis, detecting certain levels of change, has allowed us to place 600 random sampling points in salt marsh across the entire Georgia coast, and we have largely selected the sites that we will begin to monitor in March, or even earlier for some of the aquatic species. I'm sorry I'm not mentioning those today, Rachel. Too much to talk about. Oh yeah, and I just wanted to show you again how we've calculated all of these landscape metrics as well, because we're stratifying the sites across a gradient of fragmentation and to represent the gradient of things we expect the species to be sensitive to. Okay, so now I'm going to move on to show an example of inductive modeling approaches, and I want to talk briefly. Let's see, how much time am I at? Okay, I think I'm still.
You're good. You're good.
Okay, I want to talk briefly about the construction of these species distribution models so that we're all on the same page. Generally, I use the schematic in my course to show what the general approach to species distribution modeling is, where we collect occurrence data on the species, predictors, environmental predictor layers in GIS that we think the species are going to be sensitive to. We have, that normally takes about 98% of your time, and then the rest of it just goes boom, 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 and then you get stuck in modeling hell, and you know, tweak and tweak and tweak. But anyway, then there's the different stages, and I'll briefly talk about those right now to represent where I'm at just with this schematic. So again, two major data sets are required to do this. The occurrence data, which consist, importantly, of known occurrences, most commonly, known absences, almost never, and suspected absences, or pseudo-negatives, or background points, or whatever you want to call them, depending on the algorithm you're going to use. And I'm not going to go a lot into those today, but it is one of the challenges of species distribution modeling is how to select that background or the absence data set so that you can best evaluate the model and produce a tight fit without overfitting and all kinds of stuff like that. And then, of course, our predictor data is the other major data set. Additionally, what we often find is that we start with a respectable number of samples and then start to investigate where the data came from and how old it is and who collected it and what kind of notes are available. And we start excluding locations for all sorts of reasons and end up with a much smaller number of samples upon which to build our model, which is always a little bit discouraging. Again, with the predictor layers, we collect things that are important. I've got vegetation, elevation, and frost-free days here. Obviously, in a climate model projection, we would be, in most cases, excluding some of the non-climatic variables, because if they change, you can't use those to project them forward. You can certainly use some things you expect to be static, like elevation. And then, of course, we have the attribution stage, where we overlay the points with all of those layers and generate the kind of information that we need to build a model where we have our response and all our predictors in the proper format. And of course, some of the algorithms do all this, do these steps by themselves, but many of us have spent many hours doing this manually in GIS. So then we get to the model generation stage, and we have a lot of choices, and they're growing. The different variety of approaches that we can use. We can use envelope models, like some of the early models used for vegetation modeling of climate change by LG and Box. Similarity metrics, maximum entropy is something I'll talk briefly about, which we use. Again, I won't go into a lot of detail. Regression-based models, ordination, nearest neighbors, convex hulls. Lots of approaches out there, and they all have their benefits and pitfalls. So there's been a lot of work. I just want to point out briefly the model evaluation stage. I won't talk about it a lot other than that it's really important to do a good job with model evaluation. There's lots of ways to do it. There have been a spate of studies that have come out and compared a lot of these different model types. Here's one done by Pilar Hernandez, who worked for NatureServe, who was involved in some of the early species distribution modeling workshops that I taught. And on this axis, we have prediction success. We have 4 different model algorithms, and then sample size down here. And this plot just shows how the prediction error decreases at different rates with sample size, and some models are more stable across sample size and others are not. And there's a myriad of different ways that you can look at these kinds of things, and people have. And the main point being they allow us to kind of pick and choose the models we think are going to be more appropriate for our species and consistently work well. This is another paper that came out, pretty oft-cited paper, Elith et al. In ecological ecography, I think, or ecological modeling, I don't remember. Anyway, we have an area under the curve and a biserial correlation, 2 axes that essentially represent model quality in increasing both directions. And we see that maximum entropy, boosted regression trees fall out way at the top, general linear modeling, general additive modeling, some of those other things fall out in the middle. And again, I'm not going to go into the details of what these models do, but evaluations like this give us a sense of what might work well, particularly because the details of these papers give us information about what species they're working well on, etc. And then, of course, we get to the mapping stage where we have to oftentimes determine some sort of threshold. And we can produce a suitability map that has a gradient, but oftentimes for decision making, we need to choose some sort of a threshold. A lot of these algorithms come out with logistic type response that ranges from 0 to 1. And we have to decide if greater than 50% probability of occurrence means that we're going to call that suitable habitat for the species or not. Inevitably, the majority of these are not normal distributions, and so you cannot choose 50% or 0.5 as your threshold, and so there's a whole art and science to choosing thresholds with respect to species distribution modeling as well. So in addition to the multiple climate scenarios that we have, the multiple threshold choices we have, the multiple algorithm choices we have, it's very, very common these days to use ensemble type approaches where we run a variety of situations, we average them, we look for concordance between them, and select some sort of composite output. And that's what I'm showing here for a pygmy rabbit in southeastern Wyoming, where we've produced a composite model that simply layers the models on top of each other and comes up with areas where only one model has predicted the habitat to be suitable, whereas the darker areas predict all models predict it to be suitable for the species, which gives us some confidence that those are important habitats for that species and some direction to go in terms of protected areas, etc. Okay, give you a brief example from Kyle Barrett's work on modeling amphibian response to climate change in the southeast. This was a grant that John Mayers and I got to hire Kyle as a postdoc, and he came in and learned a lot of these approaches really fast, did a fantastic job, as evidenced by his marketability in this climate. In any case, I'm going to give this example of inductive modeling from Kyle's work. So we have so many problems, so little time. The Southeast represents one of the world's most diverse regions for amphibians. Of course, the Appalachian area is a global biodiversity hotspot for amphibians. There's nearly 200 species, over 50 of them are endemic. Climate change is likely to result in a loss of climatically suitable habitat for many of these species. So how are we going to get at this problem? And what species do we choose? In this particular case, Kyle talked to a lot of state herpetologists. Basically got some expert opinion to select the species. And our goal in this project was to identify results that might help wildlife managers, particularly in current protected areas, to decide on management options. We had, I think, 21 original species for this project. I can honestly tell you I don't know any of those, with the possible exception, someone can correct me if I'm wrong, is this, and the AA one there is, I think, the green salamander, just because I scanned across it several times. So again, my lab is very collaborative, and I do that on purpose. I'm a terrible natural historian, and that's almost the first thing I always tell people. So there's a few things I know about fish, and give me a salamander in a tree, and I can't tell you what it is. But I can bring some of the modeling tools to bear on things. This is generally the location of some of these species. We have several in the southern Alps, a couple in the Piedmont, several coastal plain species, and then some that are generalists. So to build this correlative model, we started with locations where the species have been observed, generated thousands of pseudo-absence locations because we were going to link what was known about what bioclimatic variables influenced these suite of species from literature studies with some of the climate scenarios to model them using maximum entropy. The maximum entropy is a little bit of a black box approach. It's really, really easy to use. Any of you could probably learn it in a day if you sat down to do so. Jeff could probably speak to that. Over there. You can make a lot of bad models using MaxEnt as well, so do so with caution. Maximum entropy employs several different algorithms internally: threshold, logistic, and other types of responses. The number of responses you can use is limited somewhat by your sample size, but a lot of those controls are set internally. Generally, what it does is it detects a signal from noise, which is why it uses the suite of background points. It's basically looking for the sort of minimizing the difference between the maximum entropy distribution and the distribution of your data given the constraints, environmental constraints. So I won't go much more into MaxEnt because I want to move on to the results. So we looked at species richness and individual species projections for 2 IPCC scenarios and 2 general circulation models, yielding 4 outcomes, 3 model thresholds per outcome: conservative threshold that would restrict the suitable habitat more, intermediate one, and a liberal threshold. And then we generated ensembles of those thresholds and the general circulation model scenarios. So here's an example of identifying sensitive areas using the species richness. And again, this isn't total species richness, it's simply the species richness of the suite of species that we had sufficient data to run models, in this case 9. So we generally see, as expected, we have several coastal plain species and several Appalachian species represented in the baseline scenario. This is the B2A, which is the more conservative projection. I think it has a little less warming than the A2A scenario. If there are climate people in here, you can correct me if I say any of those things wrong. Sometimes I forget which is which because you get so used to talking about them using their letters, you forget which ones have the stronger warming and etc. But I think that was right. In any case, you can see a dramatic loss of the species richness of these perceived sensitive species in those areas, particularly in the coastal plain region. They're almost completely eliminated. And even though this graphic shows the extension of those habitats moving, in general, we're assuming that these species, because of their life history traits and their local characteristics, won't in fact be able to move, particularly because in the mountains they would have to go down in order to go up again, and we don't expect amphibians will show that kind of adaptive ability. But again, we don't know in a lot of cases. So we wanted to identify sensitive species that showed 100% of projected loss of suitable habitat. So that's just what I'm going to show right here for some of these species. The black is 100% loss of habitat for the species under each of the scenarios, and each of the thresholds, that's the MTP, F10, and MTR mean. Those are just the different thresholds, model thresholds that we chose. And in fact, for these other species here, you can see that it's nearly 100% loss for most of those. These are coastal plain species, one species in the Piedmont, and then several, 3 species in the Appalachians. And again, these are just the top species that show the 100% or nearly 100% loss of habitat. There's that one scenario down there where we see a slight gain in habitat in that particular species and scenario. But again, the story in general for amphibians doesn't look too good. Here's an example from the A2A scenario of that green salamander. You can see the original distribution map sort of outlined there, and you can see what the, and this is the ensemble model where we took the results from all of the scenarios and put them together. So the redder areas show the areas that are most likely to remain suitable or to become suitable for the species. And the bluer areas are the areas that only, like I said, one scenario or one model supported as being suitable. So again, we took this one more level where we prioritized some of the protected areas across those distributions, and we developed an index that we could apply to the different areas over which certain people would have some management authority to provide the partners with some information on these model results. For example, how much of those protected areas had agreement that there would be some important habitat there. This case where a few models suggested that the whole area would be suitable, and then the final case where several models agree that the majority of the protected area will remain suitable for the species and therefore be an important aspect of management of that unit. So in summary, coastal plain habitats shift in suitability as much as other hotspots, like the southern ash. Over half of the modeled species are projected to lose over 90% of currently suitable habitat. And again, ensembling was useful for us in terms of a tool to identify some of these potentially important areas for future protection. Okay, some acknowledgments for that one. We'll move right on. There's a lot of partners on that project. So as a final example, I want to get back to the mechanisms a little bit, talk about a few other approaches that are out there. Again, we've been talking primarily about forecasting species distribution shifts using these correlative or inductive models. They're phenomenological. They relate current distributions to these environmental variables. But they have some faults in that they can't necessarily represent physiologically what's going to occur. A lot of our observations of where those species are now may in fact be flawed in and of itself in terms of our ability to detect them and other pressures they currently have, which may or may not be there in the future. So there's a suggestion by several people that we need to go back to mechanism. We need to understand more about the physiological responses of the species, and a lot of the first papers to come out on that in recent years, even though I feel like that's where we started 15, 20 years ago, is from Warren Porter's lab at the University of Wisconsin, and some of the postdocs he's had recently published some nice papers looking at doing some mechanistic approaches, although again, they do require a lot of information that we don't have for certain species. But we're going there anyway. Particularly where we do have some information about species. And so I want to talk briefly about a project that has a small climate change component. We're just not quite there yet with the modeling, but I'll show you some of the preliminary results. This is a project, this was my first NSF project that we were awarded just before I came to the University of Georgia, and my first PhD student, or one of my first 3 PhD students, Shannon Albeck, came to Georgia to work on this project and do a lot of the modeling that ensued. This project was called From Latrines to Landscapes, Effects of Temporal and Spatial Variation in Nutrient Transport by Coastal River Otters on Terrestrial Communities. This comes from a long history of work by my colleague Merab David, who was involved right after the Exxon Valdez spill. She was a mammal ecologist in Alaska at the time, and became one of the key scientists to work on the river otters and their recovery in all the ensuing years. And so she had a lot of good behavioral and physiological information about how the otters were responding to oil, and in fact, how they were responding to influences in their environment in general. What she discovered in the process is that they have some very interesting behaviors that have some ecosystem level consequences. Particularly, coastal river otters use these latrine sites on the coastal terrestrial landscape. And these are river otters, but they're using salt water as their primary mode of transportation. But they do seek out freshwater enclaves in the terrestrial environment. So she quantified fecal deposition, and this was also a goal of our project during the time it was ongoing. And then we were going to look at the nutrient cycling and some of the microorganism and plant responses in the terrestrial community. And again, this is where we're talking about, well, I'll show you how it's related to climate in a minute. And then how that might scale up to affect the landscape in general. Okay, so here's the conceptual model. We have atmosphere-ocean interactions which affect, of course, the coastal Alaska current. The position of the coastal Alaska current has an influence on the productivity of Prince William Sound. Okay, so in general, in certain situations, we see a low influx of nutrients and zooplankton, and then river otters will forage primarily on intertidal fishes because we don't see the volume of schooling pelagic fish returning to the sound in the numbers that normally occur. And in this situation, otters will forage solitarily, and they have dispersed and low levels of latrine use. In the alternate situation where you do have large schooling pelagic fishes returning to Prince William Sound, otters will form social groups. They will use latrine sites very heavily. Social groups will dominate. And of course, the pattern of this affects the spatial and temporal variation in that marine-derived nutrient transport to the coastal terrestrial environment. So we wanted to look at what the consequences of the declining fish populations in Prince William Sound might have. Here's just a graphic from some prior work from Merab and her colleague Gail Blundell, showing across the months of the year here river otters' minimum group size, and you see one there sort of in the lower middle. And then we see a lot of the important fish species that they rely on, the schooling pelagic fish that are often coming in to spawn. And you can see salmon availability plotted there, and we also have juvenile herring and I think sand lance, or I have it up there, or capelin. Oh yeah, it's capelin. In any case, you can see when the schooling pelagic fish begin to dominate, otters start forming social groups, and then of course their behavioral observations show that they will then start using these latrine sites en masse because they use them for social communication. It's the first time this has happened in a while, where it converts my symbols, but they still sort of mean what they mean. They're varying in size, if you can't see that. Those are supposed to be dots. The X's are correct. What we see here are latrine sites along the coast, and the years should be on there somewhere. In any case, it shows two separate years. And again, there's variability in the nutrient deposition on the coastal terrestrial landscape at the latrine sites, depending on the dynamics in a given year. Okay, so we wanted to use an individual-based model to do this. Why? Basically, because individuals are the building blocks of ecosystems. It's a natural scale in which to measure ecological interactions. Is there any bacteriologists in here? or atomic physicists. Okay, individuals are the building blocks of our ecosystem. In any case, it responds to internal and external stimuli, so we can model this adaptive behavior, and it leads to our observation of sort of an ecosystem level response, or at least a population level response. And of course, it's somewhat easy, although sometimes expensive, to measure and predict the behavior response of an individual, and then by modeling all individuals, you can get some emergent properties for the population. So we modeled this in a linear framework. This was one of the sort of innovative aspects of this project. Instead of a traditional two-dimensional cell-to-cell individual-based or agent-based model, we built a network-based model where the animals were allowed to move along the coastline, and then according to what we knew about how much sea they would actually traverse, they had certain crossover points they could use to cross bays, etc. So this is our general study area. We had a camp for 2 years in the mouth of Herring Bay right here, and Prince William Sound is up to the left. I think that's all I need to say about that. So what is the otter's decision process in this model? Basically, they have to move on an hourly time step, okay? They move based on a normal distribution of observed otter movements, where the model randomly samples. That's typically how these models work, and I won't go into a lot of other detail, but there's a distribution assigned to each of the mechanisms driving the otter decision-making in the model, and we simulate by drawing from those distributions and giving an otter in an individual time step a particular given response, which is determined by its current state, etc., etc. So it can detect ascent. Okay, so it's going to detect nearby latrine sites, depending on how and when those latrine sites have been used by other otters. And it does that based on distance and the feces age on that site. It can switch to an inactive state or a resting state. The otters were observed to rest actually quite frequently and sometimes for long periods of time. And that, of course, depends on how many hours they've been active and whether there are fish schools nearby. They can switch to an active state based, again, on how many hours they've been inactive and whether there are fish schools nearby. And then, of course, they defecate, the key to the whole modeling, the nutrient transport. And that, of course, depends on how long it's been since they last defecated, whether they've been on a fish school recently, and, of course, whether the habitat is suitable or whether there's a latrine site nearby or whether they have to move again in order to reach one. This is just a schematic that I won't go through for that IBM, where we model each otter at a time step and go through each of those decision-making procedures that I just outlined. So how well did we do? And I have to make the point that these responses I'm going to show you had absolutely no input on the parameterization of the model. So the neatest thing, which you'll see at the end of this, is that just using what's known historically about the behavior of these otters and how they're responding to certain cues, we were able to do pretty well at reflecting emergent patterns observed in the landscape. We observed 39.9% feces with pelagic fish, 36.6% feces in the model contained pelagic fish. 43% of males and 32% of females switched their diet to pelagic fish in the model, 38% for males and 35% for females. There were approximately 2 otters observed per group on average, although it varies and they're sometimes in group sizes in excess of 8. And then our average grouping in the model was 2.5 otters. And again, this is all for our what we call our baseline scenario. We did a sensitivity analysis and some other things. 9 to 11 was the maximum group size ever observed in Maroff's prior studies, and we had a maximum group size, I think, just in one simulation of 14 animals. Males within social groups significantly more than females, and we saw, again, males grouping up more often than females, depends on their reproductive state. Females occurred in mixed gender groups, at a rate of 78%, and males at a rate of 37.5%. In the model, females occurred in mixed gender groups at a rate of 80%, and males at a rate of 26%. So we were really happy with this, and in fact, one of the first times we ran the model, the actual number of species estimated on the landscape in our year of sampling was literally within like a few hundred, and we're talking thousands and thousands of species. It was actually within a few hundred of what the model simulation predicted, which kind of blew me away, and it's surely an anomaly. So as we continue to work on this, I'm sure the results will just get worse and worse. Anyway, we did a brief correspondence analysis. The most important part of which is really sort of what these axes mean. Along here we have defecation rate. There's other stuff going on here, but I wanted to kind of simplify it. Generally over here represents larger groups, and this is smaller groups. We did a scenario where we had informed fish schools. So fish schools school up at certain types of marine habitat, and we sampled from a distribution and again put those fish schools out on the landscape and made them dynamic. But they were informed. They were using habitat that the fish schools and size ranges that we had observed in reality. We randomized them, and then we did no schools. And in generally what we're seeing here is that in the no schools scenario, which again is going to be the extreme of if schooling fish populations continue to decline, we see smaller group formation and a decline in the defecation rate observed at the latrine sites on the coastal landscape, and so then a decline in nutrient deposition. So then the question is, what is going to be this ultimate effect on the landscape pattern? And again, we're not quite there with the modeling yet. I actually did a brief agent-based modeling exercise using the latrine sites as individuals. The latrine sites then got visited by various groups of otters. I did this actually to develop the proposal in the first place. So what we see here is Delin-15 nitrogen of spruce foliage, which has been an indicator both in the Yellowstone Lake ecosystem and in Prince William Sound of deposition by river otters in a latrine. And what we see along the top is heavy latrine use with no warming, heavy latrine use with warming being the one that's sort of coming down there, and then your average non-latrine site, which has very, very low Delin-15 values because they're not receiving any enriched nutrients from the aquatic environment. And again, looking at it in terms of heterogeneity, we look at the coefficient of variation here of Delin-15, and we can see that with warming, and we see a continuous decline in the heterogeneity of that nutrient deposition on the coastal landscape. I'm going to quit. That was another brand new result, but I'm not going to talk about that. In summary, we have a lot of useful approaches available to us to look at vertebrate species response to climate change. Our goals in the NATE lab are to apply the appropriate tools depending on species, environment, state of knowledge, management need, etc., to innovate with respect to application of unique spatial tools, and of course to geek out on complex but very informative spatially explicit models. So with that, thank you for bearing with me for almost an hour.